My name is Brian Riley, and this is an introduction to arthropods. The purpose of this presentation series is to introduce students to the concepts and terminology relating to arthropods and to provide a basic background for identification of major terrestrial arthropod groups. Before we start talking about arthropods, I'd like to talk a little bit about how we identify, organize, and describe life forms in general. And these things fall under the topic of systematics. Systematics is the study of the diversification of living forms and of the relationships among living things through time. Systematicists, or scientists who practice systematics, seek to do a number of things. First, they seek to provide names for organisms. They seek to describe them. They seek to preserve specimens of them. And then they seek to classify them, provide keys for their identification, and supply data on their distributions. Next, they also investigate their evolutionary histories and consider their environmental adaptations. The first four of these items fall under a subfield of systematics known as taxonomy. And the second two, or the last two of these, fall under a subfield of systematics referred to as phylogenetics. First of all, taxonomy is a science of grouping organisms and giving names to those groups. Groups that are dealt with in taxonomy are referred to as taxa, or singular, a taxon. A taxon is a group of one or more populations of organisms thought to form a unit. Taxonomists commonly remain at odds over what organisms do or don't belong to a given taxon, and the criteria used for inclusion in that taxon. A taxon is not necessarily a monophyletic group, or sometimes you'll see them called clades, but the goal of modern systematics is to create taxa that are monophyletic. Monophyletic groups of organisms contain a common ancestor and all descendants of that common ancestor. You can see this pictured in yellow with the common ancestor demarcated here. A few other important terms here are paraphyletic group, which you can see in blue down here. A paraphyletic group contains a number of organisms and a common ancestor, but not all the descendants of that common ancestor. So you can see the yellow group is not within that blue group there, and therefore it's paraphyletic. The last term that would be useful to have here is polyphyletic, or a polyphyletic group. And this is pictured by these red blob up top. The night-active primates in this particular tree. A polyphyletic group is defined as multiple organisms in a group without a common ancestor within that group. So the common ancestor of lorises and tarsiers in this case would be also down here at this arrow, but it's not included in the group of night-active primates. We've talked a bit about taxonomy at this point, which is the branch of systematics that provides names for organisms and describes organisms. Now we're going to talk a little bit about phylogenetics, which is the investigation of these organisms' evolutionary histories. Phylogenetics is the study of the evolutionary history and relationships among individuals or groups of organisms. Phylogenetics uses heritable traits, such as DNA sequences or morphological characters, to infer phylogenetic trees, as pictured down below. Let's have a look at this phylogenetic tree. This tree, like others we will look at in future presentations, is considered a rooted tree. This means that at the base of this tree there is a root, which represents the common ancestor to all taxa within the tree. At each branching point on this tree, there is an internal node. These nodes represent the common ancestors of the branches that come from that node. At the tips of the tree are another set of nodes. These nodes have no further children, i.e., there are no branches coming off of them, So therefore these nodes are called leaf nodes. Each branching point on this tree represents a difference in one or more traits. Which brings us to a discussion of the terms trait and character. A character is an observable feature of an organism, while a trait is a distinct variant of a character. For example, eye color would be a character, for which brown would be a trait. Shown here are a few important concepts regarding phylogenetic trees and the traits used to build them. A plesiomorphy is an ancestral trait observed at the root of a given tree. Because plesiomorphies are ancestral traits, they are not indicative of relationships within a tree. An apomorphy is a derived trait observed in some unified section of a tree that does not include the root. One type of apomorphy is an odd apomorphy. An odd apomorphy is a derived trait that is unique to only one given taxon. A second type of apomorphy is a synapomorphy. A synapomorphy is a derived trait shared by multiple taxa and their common ancestor. Synapomorphies distinguish monophyletic groups from other organisms. Finally, a homoplasy is a trait derived separately in two taxa and not observed in their common ancestor. Now that we have a bit of an understanding of these definitions and concepts, let's talk a little bit about how we actually identify organisms. One of the most common ways to identify organisms is through the use of identification keys. A key is a device that aids in the identification of organisms. Keys use traits for different characters to separate organisms into different groups. The user of a key answers a series of questions about characters of a specimen being identified. Typically, each step in this series reduces the number of potential taxa that the biological entity being identified could be. One common type of key is a single-access key. Single-access keys have a single question to start with that leads to another question, to another question, to another question. The user goes through answering question by question until they finally get to an identification. There are two varieties of single-access keys. First, there are dichotomous keys, at which there are only two options at each step. Second, there are polytomous keys, in which there are more than two options at some or all steps. Single-access keys are the most traditionally used type of key. Single-access keys are readily available for many taxonomic groups, and there are many keys for higher level groups, for example, order or family, that have been well tested over the decades and are now somewhat idiot-proof, if you will. The main problem with single-access keys is that they do not allow for readily skipping steps when the information to answer a step is not available. For example, with the key on the right, what if you don't know if your organism has a backbone? You're just kind of stuck at that first step. With single-access keys, you're stuck using the characters sequentially. The second type of key that's been developed to deal with this skipping problem are multi-access keys. Multi-access keys allow for skipping of uncertain or unavailable characters. However, multi-access keys are not as readily available for many groups as single-access keys are. Here is an example of a multi-access key for the grasshoppers of the western United States. At the top left are characters available to be chosen. Below that are characters that have been chosen by the user. Bottom right are organisms that have been weeded out from the list using the chosen characters. At the top right are species that have not been eliminated as the identity of the specimen being keyed. After this discussion, a few questions remain. First, how do you identify organisms for which there is no key? And second, how do you know that the name you've keyed your specimen to is correct? In other words, how do you know you followed the right path in the key? One way to deal with these issues is identification by comparison. First, you can compare your specimen to other physical specimens that have been collected and identified previously. A second option is to compare your specimen to images of specimen, such as those digitalized and provided online in various databases. A final option is to resort to using species descriptions. The problem is that species descriptions sometimes vary from lengthy descriptions complete with figures and pictures to a few lines in Latin. The means used to identify a specimen really vary depending upon the resources available to an identifier.